certain tissue is lost from your body, that's the end of it. Heart attack, you lose cardiac muscle forever. Stroke, good luck trying to grow new neurons. An example that I am very familiar with as a training periodontist is bone loss as a complication of gum disease, which is a very common condition. Have you ever imagined being able to regenerate the tissue lost from your body? I have, and I am here today to shed a light on this matter. Stem cells are cells that have the ability to transform into any tissue in the body, provided they are given the right kind of stimuli. Stem cells transplantation has been widely explored for tissue regeneration. However, despite showing promising and reliable results, they do not come with zero complication. Stem cells transplantation may trigger host immune response, hence causing rejection of the implanted stem cells. Other risks include increase in the probability of tumor formation as well as transmission of infection. And the worst one of all, cell-based therapy is technically very difficult because the cells are very delicate to handle. So how can we overcome these limitations? This is where my work comes in. While still using stem cells, I do not transplant them to provide tissue regeneration. Instead, I grow them, which I obtain from extracted premolative in a special solution in the laboratory. After a certain period of time has passed, this solution will contain all the proteins produced by the cell, termed secretomes. Secretomes from various types of stem cells are able to stimulate growth and transformation of affected tissue in gum disease into body tissues in the lab. However, translating these findings into a practical solution to be used in the clinic is a new challenge. As you can probably guess by now, since secretomes are essentially a mixture of proteins, they are readily destroyed if given just like that. Thus, the effect of secretome can be maximized by combining them with a delivery agent to the affected tissue. For this purpose, I decided to use carbon nanotubes. Do you still remember those mechanical pencil leads that you used in school? Shrink them by a factor of 1 million and voila, you get carbon nanotube. They have been repeatedly shown to be able to penetrate into the tissue and be retained there for prolonged periods. Once combined with the secretomes, they can ensure that secretomes are delivered deep into the desired tissue and is retained there for an adequate amount of time for regeneration to occur. Therefore, don't you think it is time to switch to secretomes delivery for tissue regeneration? Remember, we don't need the cell therapy, what we need is only the products of the cells. Thank you.